Bismillah ar-Rahman ar-Rahim. Now we call for the opening of session 6. Meet the international guest speaker, and we have an online presentation with Professor David Gozal. Professor David Gozal is the Dean at John Edwards School of Medicine and Vice President for Health Affairs at Marshall University, West Virginia. He's the past president for the American Thoracic Society and the past member of the Board of Directors of the Sleep Research Society. And today, Professor Gozal will give us his presentation entitled Sleep Apnea and Cancer, Decrypting Complex Interactions. Good morning, Professor Gozal.
Good afternoon to everybody. Good evening. First of all, thank you to the organizers for the very kind invitation. I'm delighted to be present virtually. And let's hope, inshallah, that in the future I'll be able to come in person and meet all of you and engage into what I hope will be very meaningful discussions and certainly have a wonderful taste of all your wonderful community and your country. My topic today, thank you. My topic today is really focused on something that we started about 15 years or so ago. I'm going to go deep a little bit into some of the biological aspects in order to show you not only the plausible biological elements that are certainly attest at the interactions between sleep apnea and cancer, but also to show you elements for which we are still working on in order to define opportunities of personalized medicine in the context of both sleep apnea and cancer. I have no conflicts of interest to declare related to this presentation, and I am sure that all of you are clearly aware that sleep apnea, OSA, is bad for you. This is a study that we did in about 3.5 million people. In gray, we show the normal controls, people without sleep apnea, and we show the frequency in blue for men and in red for women of arrhythmias, depression, ischemic heart disease, congestive heart failure, stroke, and other conditions. And the important part here is the fact that as the age changes, depending on the decades of age, every 5 years, you can see now that there are changes in the overall impact of sleep apnea as far as the prevalence. As would be expected, the younger you are, the effect may not be as pronounced, for example, in arrhythmias, but it can be quite already substantial in areas such as depression, particularly among women. So we know that sleep apnea has had a very significant impact on both Alzheimer's, cardiovascular and metabolic disease. And that was where the field was until in 2008, we saw this paper. This paper was published by Terry Young on the Wisconsin longitudinal cohort study. This is a study of normal individuals living in the community, about 1,600 or so, who were followed every 5 years with sleep studies and with everything else that relates to health. And Dr. Young published a very interesting element related to mortality and showed that as the severity of sleep apnea increased, the survival was reduced. Well, this was very important because this was in the general community. And the comments that she made in the paper were that cardiovascular was an important component of this mortality. But interestingly, in the text, she mentioned cancer, that there was more cancer, but yet she did not really focus or present anything in the tables. An investigator, that was the senior statistician in the group for Dr. Young's study, Javier Nieto, happens to be a very good friend of mine. And when I read the paper, I called him and I said, well, what's happening with cancer? You mentioned this in the text, but you didn't comment any further. And so since then, there was a second follow-up, this time 22 years follow-up on 1,500 subjects. And I asked Dr. Nieto to actually take a look and see whether by looking at the severities of sleep apnea, he could find issues related to all-cause mortality, but also on cancer mortality. And you can see that when you reach moderate to severe sleep apnea, there is a major increase in the, as shown here in the bottom, as a major increase in the mortality related to cancer, something that had not been reported before. And this potential association between cancer and sleep apnea became for the first time an hypothesis that was meritorious enough to go and explore. And this is the data now shown in a Kaplan-Meier curve showing that the survival related to cancer, so survival-free mortality, cancer mortality, shows that as the severity of sleep apnea increases, the mortality is, or the survival is reduced related to cancer. This was really a fundamental observation that resulted from some comments made in a paper that was really focused on mortality and survival related to cardiovascular disease. In order to further explore this, we then, as part of the Spanish sleep group, which I'm one of the founders, decided to take a look at whether we could identify similar phenomena in a population study. This is a study of 5,500 or so patients with multiple sleep centers participating. You can see their labels and their location. And of these, what we found is that from this cohort, there were 369 deaths over a period of 4.5 years of people with sleep apnea. But interestingly enough, 25% of those deaths were related to cancer. So this was now for the second time we found a substantial association between mortality and cancer in a population of patients with sleep apnea that could also show that there were significant relationships to the hypoxic burden of these patients. You can see that the mortality from cancer in normal controls without sleep apnea was low. And you can see that the mortality of this patient population that had started the study without knowledge of having cancer was dying of cancer within 5 years, much more than people that did not have sleep apnea. So we did then a study in the United States of about 1.7 million people. The idea was that we would have 3 groups: a group of obstructive sleep apnea, shown here as OSA, a group of comorbidity matched control group. In other words, they would be matched to every patient with OSA would have the same age, same gender, and all the other comorbidities, but not have OSA. And then a group, because of the location and potential exposures, a group of demographically matched control groups living in the same residence that would also match by age, gender, and the residential region. And each of these groups consists of a little bit over 1.7 million people. So this is a very large data study. I don't expect you to see anything from this table. This is just to illustrate a very large cluster of very large data sets. But the important part was that then we were able to look at first prevalence and mortality related to cancer as it relates to specific cancer conditions. So for example, bladder, kidney, ovary, breast, liver, et cetera, And interestingly enough, there were 2 major elements that came through, which are summarized on the left, is that 3 cancers of all the cancers really gave a very strong signal that indicated that sleep apnea was definitely associated with kidney cancer, pancreatic cancer, and melanoma. Now, there were doubts as to other cancers, and this was something that we could not resolve based on the design of the study. And so this left us with a suspicion that certain types of cancer at an organ level are going to be more likely to be affected by sleep apnea, but doesn't tell you much about within every organ, whether any specific type of cancers within the organ may be susceptible, but the others not. So this was, of course, the beginning of an exploration at the epidemiological levels for cohorts that were not designed specifically for this purpose. We followed up with a study in the state of Washington the University of Washington Sleep Center. We identified a cohort that was a cancer cohort related to the cancer registry at the state of Washington, and then identified patients that had sleep apnea, were not selected or not selected. And on the right, what you see is that different types of cancer show the yellow line indicates the equipoise. In other words, if it's your below, there's no evidence that sleep apnea will affect your probability of cancer. But if you're above the line, it would indicate that in this population, there is a signal that is provided as a portion of the variant is explained by the increase by the presence of sleep apnea and its severity. And you can see that now colorectal cancer, corpus uteri, lung, melanoma again, prostate, and thyroid appear when in fact in the previous large cohort we didn't see it. So you can see that there's variability that is geographical, there's variability related to the population, and most likely we assume that there will be variability, very significant variability related to the types of cancers within any specific organ, because again, all these are related to all the data that I'm showing you is related to organ, not specifically to a certain type of cancer. Now, since then, there's been a very large number of studies related to incidence of cancer in relation to the presence of sleep apnea. I'm not going to show you, just wanted to give an example of all the data that has emerged in a very, very flurry of data sets that were never really designed for this purpose, but nonetheless include a very large number of patients, as you can see, all the way from our study with 5.6 million, but others with 10,000 people, 200,000 people, etc. And the important part is that most of them focused on overall, but with an organ based, when possible, based on the large size of the cohort. And the summary of these are that some, for example, a Canadian study, a Spanish study, a study from Norway, and others gave us a signal that was positive, while others, such as the one from Canada, our own, in a very large study, etc., gave us a signal that was very borderline. Yes, for certain types of cancers, not for others. So the incidence risk was very variable on the basis of cohorts that were historical cohorts that had not been designed to study the incidence of cancer as a function of sleep apnea, and with a lot of multiple confounders that may or may not be identified. So the summary of these studies are that one, there's an inconsistent overall cancer site association, there's heterogeneity across cancer sites, and that combining cancer sites obscures even further these differences, but that there are consistent associations across all the studies that examine this particularly for melanoma, pancreatic cancer, prostate cancer, and kidney cancer. And so, because these are cancers that are quite frequent, of course, us as pulmonologists were interested in lung adenocarcinoma, but I'll leave it for a second. And what focused on was, because of the consistency of all of these elements, we decided to explore melanoma. One, because there's a very affordable and accessible model in animals of melanoma cancers, and two, where we could explore models of sleep apnea and then decipher, without all the complications of the environment, food, etc., and confounders such as body weight, smoking and non-smoking, etc., we could explore this issue of a cancer type being susceptible to sleep apnea. But before that, we designed a study where, for the first time, we were going to focus on melanoma, but do this in a clinical way that was targeted to partner with dermatologists. So we collaborated with a group of dermatologists in the Spanish sleep group, identified 476 patients that were initially screened with confirmed cutaneous melanoma. They were all needed to be 18 years of age and below and above. All other melanomas, if they had sleep apnea known or they had previous CPAP treatment, were excluded. And at the end of the day, every patient that was identified with melanoma, we ultimately did a sleep study within a very short period of time. And at the end of all this, 443 patients with melanoma underwent a sleep study. in which we had very extensive characterization of their biological markers of aggression of melanoma and looked at the presence of sleep apnea in this population. And what we found is that whether we looked at the oxygen desaturation index or the AHI, the markers of aggressiveness of melanoma were all significantly higher in patients that had sleep apnea as opposed to those who did not have sleep apnea in this cohort. So for the first time, we identified that a group of cutaneous melanoma patients that had never been diagnosed with sleep apnea, a subset of these patients had sleep apnea, and more importantly, as the sleep apnea became more severe, the likelihood of having components of very high aggression and ultimately bad prognosis was higher in those patients with sleep apnea who had melanoma compared to those who had melanoma, similar type of melanoma, but nonetheless did not have sleep apnea. So this was for the first time a study specifically targeting a large cluster, of course, of melanoma. There are many, many variants. And of course, we'll touch a little bit on this in the end of my presentation. But nonetheless, for the first time, we saw this very strong association where the severity of sleep apnea accounted for some of the aggressiveness markers of patients with melanoma. So before we start really understanding what we're trying to do, what we need to understand is that there are elements that come from cancer, elements, the biology of cancer that is being investigated, and others that come from many of our studies of multiple groups on sleep apnea. And what we need to understand is that there's a common pathways that are being identified. All these, of course, are very important on the right side for cancer aggravation, in other words, what makes cancer worse, carcinogenesis, proliferation, hypoxia, angiogenesis, and so on. What we know that intermittent hypoxia and sleep fragmentation are very important to the phenotypic markers and phenotypic expression in the clinical setting of patients with sleep apnea. But what these two conditions do essentially is to enhance DNA mutations, activate hypoxia-inducible factors, create oxidative stress, lead to inflammation, deregulate the immune system, increase the sympathetic system imbalance, and promote vascular endothelial growth factor that then leads to a potential area for both increased growth of the tumor and also for metastasis. So you can see that there's a very large overlap between these two that is reflected in these elements, and therefore we sought to start studying this in an animal model. To do that, I had developed in my laboratory a model of sleep apnea with intermittent hypoxia. We developed another one with fragmented sleep that did not have intermittent hypoxia. I will focus today only on the intermittent hypoxia, although we have quite a bit of data on all the other elements, always as a normoxic control, in other words, no alterations. And what we can really identify is different cancer models and then look at the outcome in a very controlled setting. So the idea behind it was that we would create intermittent hypoxia, create this oscillation in the tumor at the beginning by injecting a tumor and causing the tumor to be very standardized, and then take a look at the effects on angiogenesis, on necrosis, on DNA damage, on the changes in the immune system, and how all of these lead to increased or changes in the malignant properties of the tumor. And so one of the first studies that we did was to take that has now been replicated, and in 2019 we published a very extensive work to look at the first element of lung cancer. I'm going to go back to melanoma, but I wanted to focus on lung cancer since we are pulmonologists. And what we did was to do in vitro. This time we took cancer cells, human lung cancer cells, different types. You can see 4 different types of cancer cells. These are all either adenocarcinoma or non-oat cell carcinoma and non-small cell carcinoma, and expose them to either normal or to 2 types of intermittent hypoxia, more severe one, more milder one, or sustained hypoxia that would be in between. And you can see that for each of these tumor cells, these are all human cancer cells, very well established, for which many of the studies on pharmacotherapy and so on are done, you can see that their response to sustained hypoxia or intermittent hypoxia of different severity is very different. For example, if you take H522, which is an adenocarcinoma, you can see that sustained hypoxia does not do much. Actually, it suppresses growth, and we don't see much of a change with intermittent hypoxia. In contrast, if you take adenocarcinoma 1437, you can see that sustained hypoxia and mild intermittent hypoxia leads to increased proliferation, not so with severe intermittent hypoxia, but conversely, an adenocarcinoma H520 does exactly the opposite. Sustained hypoxia leads to no major changes, but you can see that intermittent hypoxia leads to explosive proliferation in this case. So different types of cancers that are maybe bare or have account for different mutations may lead to very different biological behaviors in the context of intermittent or sustained hypoxia, which then may explain why the epidemiological studies that are not specifically targeting not only a certain type of cancer, but also a subtype based on their mutations, may yield very conflicting results that then do not explain the lack of association or the very strong association between one study and the next. So, as I mentioned, mice have a certain type of cutaneous melanoma that we can explore. This is what we took. We took in a group of mice, we took a known number of melanoma cells that we injected, and we exposed them to intermittent hypoxia mimicking moderate to severe sleep apnea. And this is the tumor after it gets injected. But the important part was this. If you take a look at the tumor volume or the tumor weight, mice that were exposed to intermittent hypoxia showed a much greater increase in the tumor size and in the tumor weight compared to those that were not exposed. Those were exposed to normal conditions. Second, we saw the presence of metastases that were more likely present in mice exposed to intermittent hypoxia. These metastases went to the lung, which is one of the targets of the cutaneous melanoma in mice and also in humans. And so you can see here the pigments showing in the lung. And the third, the metastatic surface altogether was much higher in mice exposed to intermittent hypoxia compared to control, which then explains why the mortality of mice with intermittent hypoxia, in other words, with sleep apnea, with this melanoma was higher than mice that did not have exposures or did not have sleep apnea. So for the first time in a biological model, we showed that indeed the presence of sleep apnea can accelerate the proliferation and metastatic potential of a tumor such as melanoma and lead to decreased survival. We repeated these studies, in this case, with a lung tumor, an adenocarcinoma of the lung. We tested it for susceptibility to intermittent hypoxia in vitro, injected it, and there were multiple features that were shown. One, you can see that the proliferation of the tumor is higher in mice exposed to intermittent hypoxia, both the tumor weight and also the presence of invasion. These are very, very well demarcated tumors that have a capsule. This is the tumor here on the left in the bottom, relative to the muscle. So you can see there's a very nice capsule. But in mice that are exposed to intermittent hypoxia, the capsule is disrupted and the tumor invades the muscle, the surrounding muscle, leading to local metastatic potential, which ultimately can become systemic metastasis. Now, this has been demonstrated by other groups, in this case, a study that was later published by a group from South Korea, indicating that mice that have lung tumor, indeed, this particular lung tumor, human lung tumor, leads to the presence of these larger tumors and larger penetration. And one of the mechanisms that we tried to see was whether inflammation was involved, either through the expression of cyclooxygenase 2, which, as you know, is IF1 alpha dependent. And what we saw is that the expression of COX2 and its downstream products of prostaglandin E2 was much higher in mice exposed to intermittent hypoxia, and that if we blocked it with celecoxib, we could actually not only have the pharmacological effect that we wanted, but also to reduce the proliferation aspects of the tumor by treating it with a COX2 inhibitor. So for the first time, we could identify at least one mechanism that could be potentially advantageous in patients with sleep apnea to add as an adjuvant therapy in the treatment of their cancer in order to minimize the impact of intermittent hypoxia or all the other elements related to sleep apnea into lung cancer progression. So from these senses of all and a great deal of a lot of studies, we decided to summarize some of our findings and identified really all the papers that had been published over 10 years or so. And the messages were as follows. One, intermittent hypoxia is most of the time in most conditions of cell tumors, cancer cells, independent of where they come from. If they respond, intermittent hypoxia for the most part is much worse than sustained hypoxia or continuous hypoxia. This is important because most of our patients don't have continuous hypoxia. They have oscillation of their hypoxic burden, and that is therefore suggests that many of our patients with chronic lung diseases that lead to hypoxemia may have these ultimately oscillations in their saturations, which then may lead to not only the onset of cancer, but also most importantly to its proliferation and metastatic potential. Two, that there were two transcription factors that seemed to be very important because the majority of the genes that are regulated by these two transcription factors, hypoxia-inducible factor and NF-kappa B, seem to be involved. Third, that there's clearly evidence of immune deregulation, both the increased activity of T regulatory cells, reduced capacity of cytotoxic CD8 positive lymphocytes, the reduced ability of natural killer cells, and that there was a transformation of macrophages from the pro-inflammatory macrophage to the pro-tumoral, in other words, favoring macrophages that support the proliferation of tumor cells within a tumor. Not only that, we could also see that tumor cells became less differentiated, which is not good, as you know. The more stemness they show, the more ability to go to de-differentiate, the more the tumor aggressiveness becomes bigger, the more the ability to induce metastatic and to be resistant to therapy is more likely. And again, not all tumors and not all variants of the same tumor respond to the same intermittent hypoxia. The only thing for which we only not have enough information is whether, if you have sleep apnea, will that make you more likely to develop a cancerous transformation just without any other elements involved here. We are doing some experiments at this point, but I don't have a conclusive result. Now, I want to switch gears a little bit and tell you about exosomes, because this is an important marker that we may use in cancer patients or patients with sleep apnea to identify potential targets for the treatment. Now, exosomes are very small particles, about 50 to or 60 nanometers to about a maximum 100 nanometers. We produce about 4 billion every day. We produce about 4 billion exosomes every day. They circulate in every fluid. All cells produce it, particularly when they get activated. And the exosomes have a very, very, very well preserved content, what we call cargo, which may have a variety of markers on the surface. It's kind of how do they dock in with other cells in order to communicate between cells. And inside, they have all these mediators, including microRNA, DNA, messenger RNA, etc. And so we can really learn a lot from both the content, the cargo, and also the surface markers in order to identify the targets and how these cells will go, to which cells they will go, and where they come from. This is how they look. This is on electron microscopy. You can see very nicely the size of these small particles in plasma, in CSF, in urine, in saliva. Just take any fluid and you will find exosomes that then can be released from a certain cell and then go to a target cell and very specifically bind to it and tell the recipient cell or the target cell what to do, how to change, how to develop good or bad or whatever it is. We can track them and we can see whether these cells, so we can take human exosomes, we label them with red here. These are cells that are melanoma cells that we are growing. And then when we take the exosomes and deliver them in a medium to these melanoma cells, you can see that they incorporate into the cells. So the melanomas, or all cells, will incorporate exosomes coming from the plasma, coming from any other fluid, and can incorporate then the message and transform the message into specific instructions to how to change their behavior. And so the first study that we did was to take tumors in vitro and pick exosomes from plasma of patients with sleep apnea and see whether they would change the behavior, both in an animal model and then in humans, whether they would change the behavior of the tumors in vitro by taking exosomes from patients with sleep apnea and controls and see what they would do. Or mice exposed to room air and intermittent hypoxia as models of disease. And you can see here that for mice, this is proliferation, migration, and invasion. You can see that mice that were exposed to intermittent hypoxia had worse invasion and worse migration and more proliferation than those that were not exposed to this. So suggesting that, again, exosomes derived from a mouse without the hypoxia, just the exosomes, already are giving orders to the cancer that changes the behavior of the cancer and makes it become more aggressive. We identify a set of microRNAs in the cargo, and then developed a downstream computational model in order to identify what the targets were. And as we expected, there's a lot of these targets are very important to cell regulation, to mitotic capabilities, and certainly for markers or elements that regulate the immune system as well. And this is the same before and after CPAP. So these are patients, healthy subjects, people with sleep apnea before and after treatment. We then took their exosomes and at the same time exposed two types of cells, colon cells and pancreatic cancer cells, and showed that indeed exosomes coming from a patient with sleep apnea before treatment were more likely to induce both proliferation and migration than after treatment, that there was improvement with CPAP treatment. And obviously that is important because it tells us that treatment of sleep apnea can withdraw some of the impact that the presence of sleep apnea may have on cancer, on a patient that has also cancer at the same time. So I'm going to tell you the story that I told you about these 400 and so forth, 450 or so patients with melanoma. We followed them for 5 years. And of course, there were multiple groups. There was a group that did not have OSA. They were treated according to standard treatment. And you can see that their mortality, of course, over 5 years so the survival is about 65% or so. Patients with sleep apnea that were treated for their sleep apnea, whether severe or moderate, had the same mortality as patients without sleep apnea for this melanoma. But there was a group of patients that had moderate or severe sleep apnea that decided not to get treated for their sleep apnea. And this patient had a much more severe, to the point that after 5 years, there was no survival whatsoever. This has been published in Chest and further attests to the fact that in these patients with melanoma, the presence of sleep apnea leads to an increased mortality if you don't treat the sleep apnea. But if you do, you can actually normalize them to the standard survival expectations that you would be in a patient without sleep apnea with the same type of melanoma. So this is a very important finding that for the first time shows us that interventions and treatment of sleep apnea can reduce mortality in a very aggressive tumor such as cutaneous melanoma, and that therefore maybe all patients with melanoma need to be identified for the presence of sleep apnea, and then decide based on the severity and so on, whether to treat or not to treat their sleep apnea. Now, a comment or a paraphrase here is the fact that maybe there are groups of patients that one may have different types of melanoma, I'll show you in a second, and two, which is also important, is the fact that this may open an opportunity for identifying those that are sensitive to sleep apnea, may identify unique pharmacological interventions that can be introduced as part of the treatment for all melanomas, even for those that do not have sleep apnea. In other words, can we develop drugs that will target a specific pathway that is hypoxia sensitive, and then use that in the context of cancer? Because as I showed you before, cancer and sleep apnea share many homologies in many of the mechanisms that lead to the formation and aggravation of a cancer. So this is just to show you that we wanted to identify a variety of melanoma cells and to look at mechanisms. These are patients before and after treatment for CPAP for 12 months. We extracted their exosomes and then identified a very large cluster of functions and pathways in melanoma cell proliferation and migration. These are the melanoma cells we can label. And in this time, we identified 3 types of melanoma cells. We screened for a very large number. I'm not going to show you all of these, but I'm going to ask you to think of both 1424 and 1675. They look very similar. They absorb these exosomes in the same way. They have no differences in the context of how they incorporate exosomes from patients with sleep apnea or with patients' controls. And this is the important element. When we exposed 1424 to exosomes from patients with sleep apnea, we saw major increases in the proliferation, for example. And then when we incorporated exosomes after treatment for a year with CPAP, we saw improvements. In contrast, if you take the 1675, no differences. In other words, exosomes from patients with sleep apnea, either before or after treatment, did not change the proliferation of these cells. In other words, 1424 appears to be sensitive to something that is in patients with sleep apnea, and 1675 is not. These are 2 very frequent human melanoma cancer cells, and yet look at the very difference, the almost radically different behaviors when exposed to exosomes coming from patients with sleep apnea. We, of course, did quite a bit of understanding what the cells were going to express in their microRNAs, and this is what we found. In cells that were sensitive to sleep apnea, there were huge principal component analysis, huge differences. The cells are completely modified. In other words, the targets of these cells and the way that they're constructed is so different from when exposed to normal conditions. That tells us that there are pathways that we can identify that will tell us about which pathways may be implicated in sleep apnea that leads to this behavior in hypoxia. And in contrast, when you take these 1675 that do not respond, we found very little of the cells were not very well modified, and they were essentially overlapping, suggesting we could not differentiate between before and after exposure to intermittent hypoxia. So we said, okay, these cells are very sensitive. These cells, there's no effect. The other one, by the way, 1619, it does not. So that tells me now that even in a cohort of patients with cutaneous melanoma, there might be those that even though you may have sleep apnea, you will not see an effect, which may explain all the paradox of having very large cohorts with organ-related cancers, as I showed you at the beginning of my talk, and yet they do not seem to have an association, when in fact a subset of these tumors, if they are sensitive to intermittent hypoxia or to other elements of sleep apnea, may show a signal that would not be picked by the epidemiological study. So we took mice and took 1424 human melanoma cells, the ones that are sensitive, and injected these into the tumor of these mice, as I showed you before, like I showed you with cutaneous melanoma in the mice. And then we treated these mice with exosomes from either controls or patients with sleep apnea. And you can see the tumor volume, okay? The tumor volume of the 1424 melanoma cells, they were not exposed to intermittent hypoxia, just to exosomes in vivo with plasma from patients with sleep apnea. And you can see that the tumor cells and the tumor size became much bigger, and you can see that also there are areas of necrosis that would suggest the presence of very significant aggressive tumors being developed in the context of just receiving exosomes from patients with CPAP. For the first time, we show that this has obviously implications for in vivo tumor growth and tumor proliferation and metastatic potential, and this further confirms the biological plausibility of the relationship between sleep apnea and cancer. Now, cancer is and sleep apnea is a very chronic disease. So the question is, what happens if instead of just doing a little bit of intermittent hypoxia, we expose tumor cells to a very long, repeated numbers of intermittent hypoxia cycles? And this is what the work that has been done more recently. We have interesting preliminary evidence, but I wanted to share it with you to give you further the unique elements of the biological composition and mutations that really relate to 1675 and 1424. Again, two very frequent cells, the melanoma cells that exist in patients with melanoma. So this is a very large group. We sequenced all the genome of these cells. There are two malignant melanoma. They are very similar in two of the genes that are involved with mutations, both BRAF and CDK9-2A. So you can see that they are very, very similar, but they differ. They differ in one gene in which the mutation in 1424 is STK11 and the other one is PTEN. Now, why is this important? Because STK11 is a metabolic upstream gene that controls about 300 targets of metabolism in the cell. PTEN controls transformation. So these are, and PTEN is not sensitive to hypoxia. So you can see that mutations here may lead to a variety of malignant transformations that are not sensitive to intermittent hypoxia, while STK11 a priori should be sensitive, and indeed, on the basis of the biology that I've shown you, 1424 melanoma cells, human melanoma cells, are very sensitive. So we wanted to see what happens with chronic exposure. And this is what we saw. In the 1675, exposure to 30 cycles, in other words, almost 7 months of continuous intermittent hypoxia, did not change the properties of proliferation. If at all, what it did was actually to reduce its proliferative potential. This is not the case with cell line 1424. And cell line 1424 gained proliferative potential over time to the point that it became so aggressive that it grew literally under visualization. Within less than a few hours, you had really doubling of the cells, and tremendous. We have done quite a bit of work to show you that all of this can be tested. We have tested it in animals. We've tested the cytotoxic ability of these cells. We've tested their matrix in tumors. We've tested what happens to how do they release exosomes from their own in a variety of ways. What is their drug resistance? I won't have time to show you all of this, of course, today, but I want you to understand that these are all areas that have allowed us now to define unique, specific elements of therapeutics, strategies with drugs that are in the market that had not been considered to date for the treatment of melanoma, just by the sample of the fact that we identified the mutations of these cells in patients. We identified these STK11 mutations, and if they have STK11 mutations, we're now targeting treatment not only with the standard therapy, but with added elements, that adjuvant elements that allow for a much better prognosis. And an ongoing trial that's just been started, where we, with CAR T cells, we target specific tumors that are STK11 positive mutations in order to see whether we can really improve the survival of these patients, whether they have sleep apnea or not. So there's a lot of future studies that need to be done with immunosurveillance, with patients with cancer, and match controls for long-term surveillance, site and type specific of cancers that need to be done. specific biomarkers of aggressiveness related to sleep apnea, et cetera, et cetera. I will not belabor. This is part of our ability to measure in three-dimensional space tumors that we then specifically treat. So that allows us to do longitudinal studies without killing the animal, and therefore assess how the tumor grows, whether it's receiving to treatment, et cetera, with human tumors. And so this is a lot of work that is doing in my laboratory, but I wanted to share with you these concepts that are important. So I'll summarize. One, sleep apnea clearly has an association with specific tumors. We believe that even within each organ tumor, there are subtypes of these cancers, the specific subtypes of cancer that are going to be sensitive to sleep apnea. They allow us to then, if we treat these patients for their sleep apnea, we may be leading to improved outcomes. And third, they're allowing us to now develop much more specific therapies that are adjuvant therapies for patients with specific mutations that are sensitive to the intermittent hypoxia related to cancer biology. With this, of course, this is the work of some of my strong collaborators shown here, and of course the Spanish Sleep Network with Professor Miguel Ángel Martínez Garcia, and most of the work done in the animal models done by Dr. Almendros, now at the University of Barcelona, and Dr. Fahed Hakim, as well as Dr. Lev Becker at the University of Chicago. With that, thank you very much for your attention. I'll be happy to entertain any questions.
Thank you very much, Professor David. Can you hear me? Huh?
Yes, I can.
Thank you. If there is any specific reason why certain organs are more susceptible than other organs for the sleep apnea? Or
can you repeat the question?
Yeah.
I was hearing one word of every two.
Yeah. Are there any reason for being certain organs are more susceptible to the sleep apnea or the hypoxic burden rather than other organs in developing cancer? Why, for example, melanoma and not other organs?
First of all, melanoma is a very highly proliferative cancer that can go to every tissue, but it has also predilections. It will go to liver, will go to brain, and we go to lung. Those are the three targets for metastasis. Certain organs have certain attributes as far as their matrix and their oxygen tensions that make certain tumors have predilection for those. So that's the first element that is important. Second, indolent tumors are more difficult to study the effect because they require much longer time. So it is possible that some of our lack of association in the epidemiological studies on a relatively short follow-up survival curves may not be sufficient to identify differences between the impact of sleep apnea or others. And the third is, unless you design studies that are specifically for patients with sleep apnea and then follow them for a long period of time, or do very large prospective population studies, unless you focus on a specific cancer type and then subtype these cancers, you may have a mixed salad. In other words, certain mutations that are really not affected by the sleep apnea versus others that are. And so that attests to the complexity of a field that is really only now emerging. But I think that the next generation of studies will really incorporate all of these elements and make us understand much better, one, the susceptibility of the cancer to go to what are the attributes of the cancer cells that are susceptible to sleep apnea or to sleep disorders, and three, what are the opportunities as far as therapies in order to design smart targeted interventions that add on to the specificity of the intervention and therefore improve the survival of our patients.
Thank you very much. If there is most of the work is related to the hypoxia. Is that related to the intermittent hypoxia only, or also to the prolonged hypoxemia? For example, patients with interstitial lung disease, COPD, and so on. So we see hypoxia. Are there a difference, or is it the sympathetic over-discharge in patient sleep apnea that makes us more susceptible?
Wonderful questions. Yes, things are not simple. Sleep, for example, short sleep, irregular sleep, fragmented sleep, all of these circadian changes, of course, people working night shift. So let's just take that element, and you can see that there are now very substantial work, epidemiological work, showing that night shift workers, for example, are at much higher risk, one, of cancer, and two, of adverse prognosis and mortality. Second, patients with lung diseases that have a chronic hypoxia and on which superimpose intermittent hypoxia, the interactions between the two maybe give to a paradoxical diversion. To one sense, sometimes be protective and sometimes be actually detrimental. So it all depends on the cancer type. And so you can see that the permutations need to be studied within the context of a particular interest that you may have in one type of cancers, and then one type of patients as the group of interest because of the high prevalence of that cancer in that population.
Thank you, Professor Gozel. On behalf of the Egyptian Society of Chest Diseases and Tuberculosis, my sincere gratitude for accepting to give this presentation, this valuable presentation about this hidden topic, the relation between obstructive sleep apnea and cancer. And we look forward to see you in Egypt next year, inshallah. Thank you a lot.
Inshallah. All the best to all of you, and I hope you continue to enjoy the Congress. And congratulations for now achieving 65 Congresses. Wow, that's impressive.
Hope the next edition will be with us here in Egypt, inshallah. Thank you a lot, Professor Gozel. And now we call for the end of this session, and we call for the chairpersons of the next session, pulmonary infection.